Hello, and thank you for coming to my talk today. Animal color vision, the opponency dilemma. My name is Carlay Latour, and I'm here to talk to you about the two kinds of opponencies you're likely to hear about in discussions of color vision. These are color opponency and cone opponency, and we'll also be talking about how they might be related. First off, color opponency. This is a theory designed to explain how humans experience color. Color opponency is built on the idea of unique hues. There are some colors that cannot be described as a combination of other colors. For example, on the left, chartreuse can be described as a combination of green and yellow, while on the right, red can't really be described as a combination of fuchsia and orange. According to color opponent theory, there are four unique hues: green, red, yellow, and blue. And not every shade of yellow is unique yellow. Unique yellow is yellow that doesn't have any tint of red or green in it. Every color across the visible spectrum can be described as a combination of two unique hues. For example, teal can be described as a combination of green and blue, or marigold yellow can be described as a combination of both yellow and red. But there are no colors that are both red and green simultaneously. And likewise, it's impossible to imagine a color that is both blue and yellow. And we know that unique hues are primary and that all other colors across the visible spectrum are composed of them through hue cancellation experiments. In this example, a participant is viewing an LED with a peak at 610 nanometers. According to color opponent theory, we expect this light to be composed of some red and some yellow. The participant is instructed to rotate a dial to adjust the amount of unique blue being input into this beam of light. The participant is instructed to adjust the amount of blue light until the beam of light no longer appears to have any blue or any yellow in it. Here, the unique blue is used to cancel out its opponent sensation, yellow. The amount of blue needed to cancel out unique yellow is added to this plot, which shows how much blue or yellow is in the composed colors across the spectrum. The point at which the slope crosses the x-axis is unique green, as it's not composed of any blue or yellow. The same experiment can be done for red and green as well. This final chart shows that every color across the visible spectrum is composed of two unique hues, except for the unique hues themselves, which are primary. In summary, color opponency is a theory about how humans experience color. This theory relies on the concept of unique hues to parameterize color experience. There are four unique hues, which are red, green, blue, and yellow, and color opponency was demonstrated using hue cancellation experiments. Determining if animals use color opponency should be important to people interested in visually guided behavior. Right now, understanding how animals experience color, not just detect or discriminate objects based on color, would be transformational. This theory of color experience, color opponency, so far has only been applied to humans. But if we know that animals use the same perceptual schema as humans, for example, color opponency, it opens doors for understanding how animals see the world. Now let's dive into cone opponency, a mechanism for integrating cone signals. The cone in cone opponency refers to cone photoreceptors, which are photoreceptors sensitive to a narrow portion of the visible spectrum and form the foundation for color vision. Cone opponent mechanisms are circuits which antagonistically compare cone signals from two or more cone classes. These circuits are named for the fact that they convey chromatic information, but they may also compute spatial or temporal information as well. Let's go through a simplified example of a cone opponent circuit. On the left-hand side of your screen, you'll see two photoreceptors with different spectral sensitivities. Information from these photoreceptors is propagated from left to right across the screen, passing through a horizontal cell, bipolar cell, and ganglion cell before being sent to the brain. If we record from two opponent cells in this circuit, what would we find? In the dark, with neither cone class being stimulated more than the other, we see that the horizontal cell is sitting at resting membrane potential, and the ganglion cell is firing at its basal firing rate. If we excite the blue cone, the horizontal cell shows an excitatory graded potential, and the ganglion cell increases its firing rate. If we excite the red cone, the horizontal cell shows an inhibitory graded potential, And as the inhibitory signal is propagated through the circuit, we see that the ganglion cell ceases firing. This is an example of a cone-opponent circuit, which is excited by blue, or short wavelength light, and inhibited by red, or long wavelength light. Unlike color opponency, which has only been applied to humans, cone opponency has been found in many animals. I reviewed the literature to determine which animals are known to use cone opponency in at least one layer of the retina or brain, and compiled a list of all these animals and more. From reptiles to amphibians and humans to goldfish, cone opponency is common. But what can we infer once we know an animal's cone opponent circuits? On the left-hand side, you'll see two cone opponent circuits which belong to a trichromatic primate. The circuit on the left is sensitive to blue and yellow, and it doesn't have any special spatial properties. The one on the right is sensitive to red and green and has high spatial resolution. This means that this animal will be better able to detect small red objects on a green background when compared to blue objects on a yellow background. This is very useful for a foraging trichromatic primate whose diet is primarily composed of red fruits that they must forage for among green leaves. So now that we know the difference between cone opponency and color opponency, how are they connected? Well, in humans, they seem very closely connected. There are two cone opponent channels which closely correspond to the unique hues. But are they connected in any non-human animals? Well, we'll first need to determine if any animals actually use color opponency. The starting place should be animals which use color categorization, but not just any color categorization will get us to unique hues. It needs to be a sophisticated categorization, which can determine if a color is primary or composed. Simple color categorization has been demonstrated in a few animals. One animal for which both color categorization and cone opponent mechanisms are known is the goldfish. So how does what we know about color opponency and cone opponency in goldfish stack up to humans? In humans, we have two cone opponent channels, which compare red versus green and blue versus yellow. Necessarily, humans have 4 unique hues, twice as many as opponent channels. Goldfish have many more cone opponent channels than the human, with 10 cone opponent classes. If they used unique hues to parameterize color vision the same way as humans, we'd expect them to have 20 unique hues. But as I already mentioned, we don't really know if goldfish use unique hues, but they have been demonstrated to use 7 color categories, far fewer than the 20 needed by opponency. So maybe the goldfish doesn't use unique hues? Or maybe they don't use all their cone opponent channels for color processing. In any case, there's still a lot of questions regarding unique hues in animals. So where do we go from here? If we want to determine if animals use color opponency, I recommend we expand our knowledge of color categorization in animals, and that we determine cone opponent mechanisms in animals for which color categorization is already known. I'd like to acknowledge and thank my co-PIs, Esteban Fernandez-Juricic and Alex Chubican, as well as Trevor Price for great conversations about these topics.